Welcome, students. Continuing this lesson, strategies for the enhancement in food production. Today, I am going to describe the plant breeding. Traditional farming can only yield a limited biomass as food for humans and animals. Better management practices and increase in acreage can increase yield, but only to a limited extent, meaning thereby that the plant breeding technology is the sole practice that is likely to increase the crop yield as far as the growing demand of food for the growing global population is concerned. So, in this connection, this green revolution initiated by the pioneer, Indian pioneer, Mr. M.S. Swaminathan, who was given the Raman Magsaysay Award for his contribution in the field of agriculture, worth mentions. This green revolution was responsible for our country not merely to meet the national requirement in food production but also helped us even to export it. Green revolution was dependent on a large extent on plant breeding techniques for the development of high yielding and disease resistant varieties in wheat, rice and maize. Plant breeding is the purposeful manipulation of plant species in order to create desired plant type that are better suited for cultivation, give better yields, and are disease resistant. Conventional plant breeding has been practiced for thousands of years. Since the beginning of human civilization, recorded evidence of plant breeding dates back to 9,000 to 11,000 years ago. Many present day crops are the result of domestication in ancient times. Today, all our major food crops are derived from domesticated varieties. Classical plant breeding involves crossing or hybridization of pure lines, followed by artificial selection to produce plant with desirable traits of higher yield, nutrition, and resistance to diseases. With advancement in genetics and molecular biology and tissue culture, plant breeding is now increasingly being carried out by using molecular genetic tools. If we were to list the traits of characters that the breeders have tried to incorporate into plant crops, the first we would list would be increased crop yield and improved quality, increased tolerance to environmental stress, that is, salinity, extreme temperature, and drought, resistance to pathogens, viruses, fungi, and bacteria, and increased tolerance to insect pests would be on our list too. Plant breeding programs are carried out in a systematic way worldwide in government institutions and commercial companies. The main steps in breeding a new genetic variety of crop are collection of variability, evaluation of selection of parents, cross hybridization among selected parents, selection and testing of superior recombinant, testing, release, and commercialization of new cultivars. Selection and testing of superior recombinant is the step that consists of selecting among the progeny of a hybrid those plants that have the desired character combination. The selection process is crucial to success of the breeding objective and requires careful scientific evaluation of the progeny. This step yields plants that are superior to both the parents. These are self pollinated for several generations till they reach the state of uniformity, that is, homozygosity, so that the characters will not segregate in the progeny. That's all in this video today. The rest will be discussed in our next class. Thank you.